Today, we're taking a look at a diagnostic tool that has been a quiet giant in medicine for about 50 years now. It's called ELISA, and it's basically a molecular detective. So, let's just start with a puzzle. Think about this. How can any test find one single specific molecule, say a piece of a virus or a particular hormone, when it's swimming in a sea of millions of other molecules? I mean, in your blood or saliva, it's like trying to find one specific grain of sand on a huge beach. How is that even possible? Well, the answer is a surprisingly elegant and really powerful technique called ELISA. This is the secret engine running behind so many tests we hear about all the time, from pregnancy tests to critical screenings for infectious diseases. Okay, so what in the world does ELISA even stand for? It's an acronym for Enzyme-Linked Immunosorbent Assay. Yeah, I know, it's a mouthful, but the core idea is actually pretty straightforward. It harnesses the power of antibodies, links them to an enzyme, and then uses that enzyme to create a color change that basically yells, "Hey, the thing you're looking for, it's right here." To really get how this works, you've got to meet the key players that make it all happen. Think of it as the molecular dream team of diagnostics. First up, you have the antigen. This is our target. It's the one molecule we're hunting for in that giant sea of stuff. This could be a protein on the surface of a virus like HIV, or a hormone like HCG that signals a pregnancy, or maybe even a pesky peanut protein in a food sample. Next, we have the real hero of the story, the antibody. This is our detective. Your own immune system makes these amazing little Y-shaped proteins to recognize and grab onto specific antigens with unbelievable precision. It's like a key that is perfectly designed to fit one and only one lock. And the final player is the enzyme. You can think of this as our signal flare. It's physically attached or linked to an antibody. By itself, it doesn't do a whole lot. And this is the key, when you add the right chemical, this enzyme kicks off a reaction that produces a visible color. It's the part of the system that lets us know the detective found its target. All right, so we've met the team. How do they actually work together? Well, let's look at the most common type of ELISA out there, the sandwich ELISA. And yep, the name is perfect because we are literally going to build a tiny molecular sandwich. Here's how it's done, step by step. First, you take a small plastic well and you coat the bottom with what are called capture antibodies. That's our first slice of bread. Then you add your sample. Let's say it's a drop of blood. If our target antigen is in there, it gets grabbed and held by those antibodies on the bottom. That's the filling for our sandwich. Next, you wash everything else away and add a second antibody, the detection antibody. This is the one with our signal flare enzyme attached. It comes in and binds to the other side of the antigen, finishing our sandwich. For the grand finale, you add a chemical substrate. And that's when the enzyme goes to work, sparking a reaction that makes the liquid in the well change color. Now here's the really brilliant part. This test isn't just a simple yes or no. The intensity of that color change tells you something important. It's directly proportional to the amount of antigen you started with. So a faint color means there was just a little bit of the target. A deep, dark color means there was a lot. This makes the test not just qualitative, telling us if something is there, but also quantitative, telling us how much. That sandwich method is incredibly powerful, for sure. But it's just one way to use this technology. The same basic principle of antibodies grabbing antigens has been tweaked into 4 main designs, each with its own pros and cons. So you've got these 4 main flavors. The direct ELISA is the simplest. It's fast, but not super sensitive. The indirect method adds an extra step that helps amplify the signal, making it more sensitive. The sandwich method, the one we just walked through, gives you the absolute best sensitivity and specificity. And then there's this clever one called competitive ELISA, where the result is actually inverted. The less color you see, the more antigen you had in your sample. It's like having a whole toolkit for different diagnostic jobs. Okay, we've got the mechanics down, but why does this all matter so much? Well, because this simple, elegant principle has become one of the most critical and versatile tools in all of modern medicine and biology. Let's do a quick history lesson. It was developed back in the early 70s to be a safer option than tests that used radioactivity. Right out of the gate, it was used to measure immune proteins and, you guessed it, the pregnancy hormone. But its real moment in the spotlight came in 1985, when it became one of the very first reliable ways to screen for HIV. And the tech keeps getting better, with new versions so sensitive you can see the results with your own eyes. It's really no exaggeration to say that from these beginnings, ELISA has become a true cornerstone technology. It's the reliable workhorse in countless labs and clinics, quietly protecting public health and pushing science forward every single day. And just think about the sheer range of this thing. We use ELISA to screen donated blood for viruses like HIV and hepatitis, to track outbreaks of the flu and cholera, for all those pregnancy and fertility tests, to look for tumor markers for cancer, to detect drug use, and even to check our food for allergens. The list just goes on and on. It touches our lives in ways we don't even realize. So if there's one thing to take away from all this, it's this. ELISA's incredible power comes from a magic combination of two things: high specificity, that's the ability to find that one unique molecule, and high sensitivity, the ability to find it even when there's only a teeny tiny amount of it. It truly is the ultimate molecular detective. It can cut through all the noise, all the chaos in a biological sample, and pinpoint the one single clue that actually matters, giving us an answer that can save a life, keep us safe, or unlock a new scientific discovery. For more than half a century, this elegant molecular sandwich has been a silent hero, saving and improving countless lives. It really makes you wonder, doesn't it? With how fast technology is moving, what will the next great breakthrough in diagnostics even look like?